potential hypotheses. One is that for the original oxygenation reaction is an evolutionary relic, and photorespiration was evolved to deal with that. So what does that mean, that it's an evolutionary relic? So you have to think, when did Rubisco first evolve on this planet? Yes, sir. So if we believe that photosynthesis is the reason behind the high concentration of oxygen in the atmosphere, then Rubisco evolved before there was high concentration of oxygen in the atmosphere. So it's an accident that Rubisco can bind both O2 and CO2. And it actually doesn't bind O2 that well. Remember, 80% less efficient in binding O2 than CO2. So it's really designed to bind CO2, but because the concentration of O2 is so high now that it has become a problem. So that's one hypothesis, that this was an evolutionary relic based on the time that Rubisco evolved, and there was no selection pressure against binding to O2 back then. And in the 80s, a lot of money went into trying to redesign Rubisco so that you would have crops that would not have oxygenation as a problem, and that all failed. So nobody was able to design a better Rubisco. And if you think about it, 2 billion years of evolution hasn't designed a better Rubisco, and our 10 years of genetic engineering didn't either. Another hypothesis is that this could actually be beneficial to plants sometimes. And that, you have to go back and look at what happens in the light reactions and why that could be potentially a problem when the plants have to close their stomata. So imagine it's really warm and sunny, and it's dry. So the roots are water stressed. They send hormones to the leaves. The leaves close their stomata, but it's nice and warm and sunny. So what is happening to these light harvesting complexes? Yes, ma'am? They're being overworked, lots and lots of excitation. All of that excitation energy is passed onto the reaction center, lots of electron transport. As you go through electron transport, you make lots of NADPH and you make lots of ATP. But to make all that NADPH and ATP, what do you need? What do you need? You need substrates. You need NADP plus and you need ADP and inorganic phosphate. Where do you get those from? What does the Calvin cycle do? Uses ATP, uses an ADPH, recycles ADP, inorganic phosphate, and NADP plus back to the light reactions. If you don't have the Calvin cycle because the stomata closed and there's no more CO2 coming in, what happens? None of this is happening. So what happens? Can any of this happen if there is no Calvin cycle? No, because if you don't have an ADP plus, then these electrons have no place to go, which means these electrons have no place to go, which means those electrons have no place to go. If you're not using the proton gradient to make ATP, you can't keep bringing protons in. There's a limit as to how much of a gradient you can build up across the membrane, and then it backs up. That means no more electron transport. But it's sunny. Electrons are not going anywhere, but it's sunny. So what's happening to those pigments? They're excited. But that electron is not going anywhere because pheophytin is not accepting any electrons and compound A is not accepting any electrons. If they're not accepting electrons, what happens to that excited chlorophyll in P700 or P680? They have to do something with that energy, and that something could be damage to the photosystems. And it turns out that the photosystem that is most often damaged very quickly is photosystem 2. And it happens to be these D proteins, the D1, D2, it's the D1 protein that's damaged very often, very quickly. Yes, sir.
If ADP is not present, how come photons can't go through the synthase? Is it because there's an active site? Or?
Well, protons bind here, and as they turn to make ATP, and if you can't do that if you don't have ADP and an organic phosphate. This is not a just simple channel. This is not an open channel like you have a sodium channel, for example. So the problem is that if you damage the photosystems, that's very costly. But photorespiration could potentially help to actually prevent damage, at least to photosystem 1. Maybe not so much to photosystem 2, although plants can do one thing. They can detach the light harvesting complex from the photosystem. So we're not going to talk about how that happens, but these light harvesting complexes are not permanently attached. They can be detached, reattached, detached, reattached. So if you don't have a light harvesting complex attached to you, then you don't get excited as a P680, because that energy has to come from the light harvesting complex. So plants can do that. They remove the light harvesting complex, protect the photosystem. You can do one more thing. Remember cyclic electron flow? What does cyclic electron flow do? Electrons from ferredoxin do not go to NADP+, they go to PQ, which means PQ does not get any electrons from photosystem 2, but PQ will then donate electrons to the cytochrome, which will then donate electrons to plastocyanin, which will then donate electrons to P700. So P700 can get excited, donate the electron to compound A, which donates it to ferredoxin, which will then donate it to PQ. And you can just keep cycling the electron like this, which establishes a proton gradient. And that proton gradient could be used to make ATP if you had ADP and inorganic phosphate. Well, gee, which reaction gives us ADP if the stomata are closed? Which reaction regenerates ADP if the stomata are closed? Photorespiration uses ATP and recycles ADP. Doesn't do anything for an ADPH, but it does recycle ADP. So if you only needed ADP, you would be okay. So if you're doing cyclic electron flow, that wouldn't be so bad. It's a total waste, but it's not really as wasteful as you might think, because cyclic electron flow allows photosystem 1 to get excited, donate its electrons, establish a small proton gradient, make ATP. The ATP is used in photorespiration, ADP is recycled, and this just continues and continues and continues. You don't make anything useful out of this, but at least you're allowing photosystem 1 to work without getting damaged. And if you happen to be lucky enough to do that before photosystem 2 got damaged and you separated the light harvesting complex 2 from photosystem 2, you can salvage both photosystems. You do keep losing carbon. So you can't do this forever, because every time the photorespiration reaction goes on Gesundheit, you do lose a carbon dioxide here. So you can manage to keep your stomata closed and do this for a little while. But if you force the plant to do this forever, it will run out of carbon. I'll die. So some evolutionary biologists have argued that this process has evolved as a safety valve in hot, dry, sunny environments, so that plants can survive closing their stomata when it's really sunny outside and protect the photosystems. They're losing some carbon, but the photosystem is very important. You save that, lose a little carbon. If the photosystems get damaged, then you have to remake all of those proteins and make all of those pigments and all those membranes. A lot more costly to do that. Think about this for a little while, and then it'll be practice questions related to this also, and so you can think about it, and then at the review session. We'll see if you have more questions. One topic we're not going to talk about is how plants increase CO2 concentration where Rubisco is, and there are examples of CO2 pumps in algae and the C4 and CAM pathways, which I wrote on your syllabus. We are not doing C4 and CAM, so we are not going to talk about C4 and CAM, but if you're interested, your textbook does have information about that. So C4 and CAM have evolved to avoid the problem with oxygen, but they have other costs. So everything has a cost and benefit depending on where you are. Some have advantages over the others. So in cooler temperatures, C3 plants, the normal plants, have advantages, but in warmer temperatures, especially when it's warm and dry, these two have an advantage. So if you're interested in that type of ecophysiology, you definitely want to read those modules. So far so good? Any questions? Yes, sir.
So the regeneration, the photo respiration, do they all take into the stroma? Or the,
I can't hear you at all.
the regeneration, the photo respiration, do they all take place in the stroma or the...
Are you talking about these reactions?
Yeah.
These reactions are all in the stroma,
Stroma.
as well as the peroxisome and the mitochondria, but the chloroplast part of it is in the stroma, which is where ATP will be used and ADP will be recycled, which is where you need it to be. All right, so plenty of time. We can start our next topic, which is cell division. Mitosis is mostly review from high school biology, so if you had it in 9th grade biology, it hasn't changed much. We will spend a little more time on meiosis after the exam. We're not going to do meiosis before the exam. So meiosis will be after the exam, and we'll do meiosis and sexual reproduction and genetics after the exam. But you are reading some articles for your quiz related to gender determination. So those you do not need to know the details of meiosis to be able to read those articles. So just so you don't freak out, on Wednesday, if we have extra time, we are not starting meiosis. I do not want to confuse you before the exam. What we are going to do on Wednesday is finish mitosis, review the article that you read, and I do have another review for photosynthesis that we're going to do on Wednesday. All right, so we'll do a quick introduction to mitosis here. Cell division and nuclear division are very important in eukaryotes for various reasons, whether it's regeneration of tissues in multicellular organisms or reproduction, or in unicellular organisms. When you divide, you are reproducing. Mitosis only happens in eukaryotes. In prokaryotes, you do get cell division and division of the genetic material, but we don't refer to it as mitosis. So if you consider eukaryotes, you might have unicellular eukaryotes like an amoeba, and then you get cell division. That's reproduction. That's how they reproduce. So that's asexual reproduction. And after we have talked about sexual reproduction, we'll talk about the advantages and disadvantages of both. If you look at development in multicellular organisms, we all start out as a single cell, and then you go through multiple cell divisions followed by differentiation to get the different tissues and organs. So you can see that in the growth and development in the sand dollar embryo, so it's the first cell division in the embryo here. And you can have cell division when you have damaged the tissues, and tissues have to be regenerated. So your skin cells are constantly sloughing off, and the outer layer is dead, but the layers below that are alive, and so you have to keep regenerating your skin cells. The lining of your digestive tract, the lining of your respiratory tract, all of those cells keep coming off, and you have to keep regenerating them. Your blood cells continuously die on a regular basis every few months. You have to regenerate those cells all the time. So all of that means simple cell division, which gives you genetically identical cells. And when we talk about sexual reproduction, we will talk